Bismillahirrahmanirrahim. My name is Dr. Saroshi Pal, and today I will talk about discovery and development of novel urease inhibitors for treatment of peptic ulcer. Here is the content which I will discuss. Firstly, I will discuss about what is ulcer, then about the peptic ulcer, Helicobacter pylori, currently available therapies, and room for ongoing research and steps involved in drug discovery process, and finally, I will conclude the whole discussion. Firstly, I will talk about what is ulcer by defining it in various ways, and then I will talk about the different types of ulcers. Basically, ulcer is a discontinuity or break in a bodily membrane that impedes the normal function of organ. Um, or it may be defined as sore on the skin or a mucous membrane accompanied by the disintegration of tissues. Here I have discussed the different types of ulcers. It could be skin ulcer, pressure ulcer, mouth ulcer, and peptic ulcer. Here you can see the in pictures how the skin ulcer, pressure ulcer, mouth ulcer, and peptic ulcers look like. Under the topic of peptic ulcer, we will discuss the different things. Firstly, I will define it, then I will discuss about the prevalence of peptic ulcer in developing and underdeveloping countries, and I will also discuss about the symptoms of peptic ulcers and causes of peptic ulcer. Peptic ulcer may be defined as ulcer in the wall of the stomach or duodenum resulting from the digestive action of the gastric juice on the mucous membrane when the latter is tender susceptible to its action is called peptic ulcer. In this slide, you can see the complete digestive system of human being, and here you can see the stomach and the duodenum in a broad picture. Or in a zoom picture, and here you can see that there is the excessive secretion which ultimately leads to the breakdown of the lining of the stomach forming on ulcer. In this slide, you can see the prevalence of peptic ulcer in developing and developed countries. Here you can see that in developed countries, the percentage of individual infected by ulcer is low comparatively to the prevalence of peptic ulcer in countries which are in developing stage. Here I will discuss about the symptoms of peptic ulcer. An ulcer may or may not have some symptoms. When symptoms occur, they may include a burning pain in the middle or upper stomach between meals or at night. Uh, mm, the symptoms also includes the bloating, heartburn, nausea, or vomiting. And in severe cases, symptoms may include dark or black stool due to the bleeding, vomiting blood, and weight loss or severe pain in the mid to upper abdomen. In this picture, you can see the different symptoms of peptic ulcer, that is the burning of stomach, lower abdominal pain, heartburn, and vomiting. Here I will discuss the causes of peptic ulcer. Basically, no single cause has been found for ulcers. However, it is now clear that peptic ulcer is the end result of an imbalance between digestive fluids in the stomach and duodenum, and ulcer can be caused by infection with a type of bacteria called Helicobacter pylori. It may also be caused by use of painkillers like non-steroidal anti-inflammatory drugs such as aspirin, brufen, or it may also because of the excess acid production from gastric mass, tumors of the acid producing cells of the stomach that increases acid. Or the risk factors which are involved in peptic ulcer are smoking, use of non-steroidal anti-inflammatory drugs, excessive use of alcohol, and unhealthy dietary habits. Recent research has indicated that over 95% cases of peptic ulcers are because of the Helicobacter pylori or because of use of non-steroidal anti-inflammatory drugs, and the rest of 5% causes for the peptic ulcer is unknown yet. In the incoming slides, we will discuss about the Helicobacter pylori, its function, urease, and mechanism of action and diagnosis of Helicobacter pylori infection. Basically, Helicobacter pylori is a gram-negative microaerophilic bacterium found in stomach. Robin Warren and Barry Marshall made the remarkable and unexpected discovery that inflammation in the stomach, as well as ulceration of the stomach or duodenum, is the result of an infection of the stomach caused by the bacterium Helicobacter. Even though peptic ulcers could be healed by inhibiting gastric acid production, they frequently relapsed since bacteria and chronic inflammation of the stomach remained. The question which kept scientists puzzled for a long time was how this bacterium survived in extremely acidic pH of stomach. The answer of the question was, in order to avoid the acidic environment of the interior of the stomach, Helicobacter pylori uses its flagella to burrow into the mucous lining of the stomach to reach the epithelial cells underneath, where there is a more neutral pH. Here you can see in picture. In addition to using chemotaxis to avoid areas of low pH, Helicobacter pylori also neutralizes the acid in its environment. It does this by producing large amounts of urease, which ultimately breaks the urea present in the stomach to carbon dioxide and ammonia. And as you know that the ammonia is basic and it actually neutralizes the stomach acid. In this slide, I will describe about the structure about the urease. Urease is an enzyme and it is a nickel containing metalloenzyme of high molecular weight, and it is present in the cytoplasm and at the outside cell surface of the bacteria. The cell surface urease is the result of cell lysis and not cell secretion, and about 30% of Helicobacter pylori cells are lysed to provide protection to the other cells. And native urease does not function below pH 3. The cell surface urease assembles itself into supraassembly which provide protection at lower pH, and more specifically urease catalyzes the hydrolysis of urea to produce ammonia and carbamate. The carbamate produced is subsequently degraded by spontaneous hydrolysis to produce another ammonia and carbonic acid. Here you can see the structure of and function of urease. Uh, Mm, and in the picture you can see the structure of urease, and the function of urease is to release carbon dioxide and ammonia. And there are some interesting effects about urea and urease. Urease was the first ever organic compound synthesized accidentally in the laboratory by Friedrich Wöhler in 1928 in order to attempt to synthesize ammonium cyanide, and after this discovery vital force theory was discarded. Here you can see that how urea was synthesized in the laboratory, and urease was the first ever enzyme crystallized. In 1926, James B. Sumner showed that urease is a protein by examining its crystallized form, and Sumner's work was the first demonstration that a pure protein can function as an enzyme, and led eventually to the recognition that most enzymes are in fact proteins, and the award of the Nobel Prize in Chemistry to Sumner was awarded in 1946. Here you can see the active site of urease. The active site of urease involves two nickel atoms, as you can see, and there are several ways to diagnose peptic ulcer. The most commonly used tests include the blood test, breath test, and stool tests. Blood tests basically can detect specific antibodies that the body's immune system develops in response to the Helicobacter pylori bacterium. Blood tests require that you drink a specialized fluid containing a substance that is broken down by the Helicobacter pylori, and the breakdown products can be detected in your blood. Stool tests can also be done in order to detect the Helicobacter pylori. The currently available therapies for the mm, to cure the infection of Helicobacter pylori is done by the drugs available in market and by some homemade remedies. Currently, no single drug cures Helicobacter pylori infection. Treatment involves about several medications for 7 to 14 days, and these may include proton pump inhibitors, antibiotics, and combination drug therapy. Proton pump inhibitors basically decreases the stomach's production of acid, which allows the tissues damaged by the infection to heal. And these are the some proton pump inhibitors I have mentioned in this slide. There are certain antibiotics also available for the cure of infection caused by Helicobacter pylori, and these are also mentioned in this slide. And combination drug therapy includes at least two antibiotics, an acid reducer, and sometimes a bismuth compound as well. And there are some combination drug therapies I have mentioned in this slide as well. And in this slide, I have mentioned some homemade remedies which could be useful in order to in order to eliminate the infection caused by Helicobacter pylori. Despite of the available therapies, there is still room for ongoing research. Actually, the currently available drugs for the treatment of peptic ulcers have side effects. For instance, they may cause headache, diarrhea, constipation, nausea, itching, and the proton pump inhibitors may interact with certain drugs, and long-term use of high-dose proton pump inhibitors may produce vitamin B12 deficiency. It may reduce acid secretion enough to cause atrophic gastritis, a risk factor for stomach cancer, and it may also mask the symptoms of stomach cancer and delay the diagnosis. The most common side effect of nearly all antibiotics are gastrointestinal problems such as cramps, nausea, vomiting, diarrhea. Allergic reactions can also occur with all antibiotics. Antibiotics double the risk of vaginal yeast infection. Consequently, there is still room to develop a new drug for peptic ulcer treatment with less side effects and more efficacy. These are the different steps which are involved in the drug discovery. They may include the target identification, target assay construction and screening, target validation, hits to leads, medicinal chemistry safety, and then it may involve the different phases. The first phase is safety, the second phase is the efficacy and safety, and the third phase is also the measure of efficacy and safety, and finally the FDA review and approve the drug. And the whole process may take 12 to 15 years and lots of money as well. Here you can also see the steps involved in drug discovery. Well, there could be two approaches to treat peptic ulcer, that is the use of antibiotics or the use of urease inhibitors. And development of drug resistance is the major problem, and moreover antibiotics have side effects as well. So the only choice is to search for novel urease inhibitors. Not only this, by discovering urease inhibitors, several other diseases can also be cured. As you can see, a large number of list of other diseases I have included in this slide. And additionally, urease inhibitors have also importance in agriculture in the soil urease inducing plant damage by ammonia toxicity and increase in the pH of soil. And enzyme inhibition is basically a key tool for drug development for a wide range of enzyme related diseases, and more than 30% of the drugs in the clinical use are enzyme inhibitors. Many pesticides and insecticides also work as enzyme inhibitors in the target organisms, and plants and other living sources can provide novel and potent enzyme inhibitors. So, urease inhibitors have been identified as a important route for the discovery of drugs for the treatment of peptic ulcer and other diseases. And in this slide you can see how enzyme inhibition assay may develop, and here you can see that how the urease inhibition assay works, and the different urease inhibitors available in literature. They may involve the substrate mimic inhibitors, or they may be mechanism based inhibitors. The substrate mimic inhibitors include the classes of thiourea, barbiturate, triazole, tothions, and azacitidine, and the mechanism-based inhibitors include the thiopheine, thiazolidine, pyridine, biscumarin. And in conclusion, we have discussed about the ulcer, the different types of ulcer, peptic ulcer, how it is developed, the available therapies, and room for ongoing research. And basically, UDS inhibitors have been successfully identified as a possible route for the treatment of peptic ulcer. Thank you very much for your patience, and if you have any question, you may ask it at my email address, that is saroj.iqbal@yahoo.com. Thank you very much again.